In this video, we will talk about Enhertu's mechanism of action. Trastuzumab deruxtecan is a HER2-targeted antibody-drug conjugate, ADC, stable in plasma that contains a humanized anti-HER2 IgG monoclonal antibody with the same amino acid sequence as trastuzumab, produced by mammalian Chinese hamster ovary cells, linked to DXd, an exatecan derivative and a topoisomerase I inhibitor. Approximately 8 molecules of deruxtecan are attached to each antibody molecule. Deruxtecan link to the antibody via covalent tetrapeptide-based cleavable linker. The action starts when the antibody portion binds to HER2 expressed on the surface of certain tumor cells. After binding, the trastuzumab deruxtecan complex then undergoes internalization and intercellular linker cleavage by lysosomal enzymes that are upregulated in cancer cells. Upon release, the membrane-permeable DXd causes DNA damage and apoptotic cell death. DXd is approximately 10 times more potent than SN38, the active metabolite of irinotecan. Upon cell death, the membrane-permeable topoisomerase I inhibitor is released from the cell and can then enter neighboring cells regardless of whether they express HER2. This phenomenon is known as the bystander effect. Trastuzumab deruxtecan is characterized by 7 key attributes: a payload mechanism of action involving a topoisomerase I inhibitor, high potency of the payload, a drug-to-antibody ratio of approximately 8, a systemic half-life of 7 days, a plasma-stable linker payload, a tumor-selective cleavable linker, and a membrane-permeable payload.